Well, hello, everybody. Thank you for joining this month's Lipid Maps webinar, which is co-presented by me and Dr. Shazia Khan, entitled A Sterol and Steroid Atlas of Mouse Brain. So my name's Bill Griffiths, and I'm based in Swansea University. My co-presenter, Dr. Shazia Khan, is based at the University of Edinburgh Clinical Research Facility. Before we begin, let me introduce you to Shazia. So Shazia is a research fellow at the Institute of Neuroscience and Cardiovascular Research at the University of Edinburgh, where she leads the mass spectrometry imaging group within the mass spectrometry core at the Edinburgh Clinical Research Facility. Shazia obtained a PhD in natural product chemistry in 2009 from the HEJ Research Institute of Chemistry at the University of Karachi. She had a spell working at Swansea University before moving in 2014 to Edinburgh University. As I said before, she works at the Centre for Cardiovascular Science. And working with Professor Ruth Andrew, she's played a key role in establishing and developing mass spectrometry imaging capabilities at the mass spectrometry core. The webinar will be about 45 minutes, allowing 10 minutes at the end for questions. So, Shazia, the floor is yours.
Thank you, Bill. Thanks for the introduction, and thanks everyone for joining us today. So today I would like to share some of our work on mapping neurosteroids in mouse brain. It is a part of big collaborative project on a Sterol and steroid atlas of mouse brain, as Bill said, in conjunction with the Swansea University Bill team who are working on neurosterol. So I'm going to talk about briefly about mass spectrometry imaging approach we developed for steroid imaging. So basically, you may know steroids is composed of 4 fused rings. Three are six membered and one is 5-membered. And most of us familiar with the steroids hormones produced by endocrine organ. But the brain is also capable of producing steroids locally called neurosteroids. And their biosynthesis occur within a specific cells, including neurons and glial cells. And their production is regulated by neurotransmitters. So they are signaling molecules and can influence the activity of neurons by interacting with different receptors and affect the transmission of signals between the neurons. So they have both inhibitory and excitatory effect. They are crucial for maintaining the proper brain functioning and also involved in various biological processes ranging from memory formation to emotional regulation, such as mood, cognition, memory, stress responses, and neuroprotection. So in human body, cholesterol is the main precursor for the synthesis of all steroids hormones, and our brain has a lot of cholesterol, as we know, and also equipped with several steroidogenic key enzymes required for the synthesis of neurosteroids. So that means our brain has capacity of producing these neurosteroids from cholesterol. Beside that, corticosterone and in mice and cortisol in human, they are also neuroactive compounds and referred as stress hormones, and are main interest of our group due to their role in regulating its stress responses and their action in metabolic inflammatory diseases and in aging. So their level is locally controlled by enzyme 11-beta-HSD1, which generate active glucocorticoid from inactive substrate. So despite the significance of neurosteroids, the spatial distribution of these steroids across the brain different region is remain largely unexplored, and how they overlaid with neurotransmitter system is also poorly understood. So the aim of this study to uncover the complex network of neurosteroid by constructing a map within the mouse brain and to create a web-based 3D neurosteroid atlas of adult brain using our in-house developed on-tissue derivatization method and mass spectrometry imaging technology. So mass spectrometry imaging is a powerful analytical technique that enables spatial localization of molecules directly within a tissue section. It can be applied to a wide range of analytes, including drug, lipid, protein, peptides, metabolites, and small molecules. Unlike traditional imaging technique, MSI does not require any specific labeling like antibodies, and it is largely untargeted and enables simultaneous measurement of hundreds of molecules while preserving histological information. So, for those who may be less familiar with the mass spectrometry, a mass spectrometer consists of three main components. And first step is ion source that convert the molecules from the samples into gas phase. And then mass analyzers separate these ions according to their mass to charge ratio, and then detector convert the beam of these ions into electrical signals that can be measured and recorded. So basically, mass spec detect gas phase ions based on their mass to charge ratio. So there are different mass ionization source and mass analyzers available, but the selection depends on nature of biological samples and research question. So how does then mass spectrometry imaging work? So in all MSI experiment, molecules are ionized from a set of raster-defined grid points on the sample surface, and a complete mass spectrum is required, which include relative abundance of each m by z, each molecule, mass to charge ratio, assigned to each pair of xy coordinate. And this process continue in a raster manner. Like this. So MSI spatially map the molecules according to their XY position and accurate mass measurement and provide a multidimensional data sets. And then software reconstruct the images by plotting the intensity of each individual ion and at each coordinate across the tissue, and then images reveal the spatial localization and relative abundance of a specific molecule throughout the sample. And to achieve a spatial resolution depends on instrument and equipment, and also laser spots and raster step size. So several ionization technique can be used for MSI, for example, MALDI, DESI, and SIMS. And in our work, we use MALDI, which is most common and widely used mass spectrometry imaging technique that employ laser beam fire on a matrix coated surface for sample ionization. And this video show you a real-time picture of how it works. So here is a raster-defined grid points, and laser is firing across the surface of tissue, which is coated by matrix, and collecting all the information from the surface, like these are the XY coordinate, and on the right side, we'll see the mass spectrum is acquired here. And there are the sets parameter, for example, raster. So that all sounds like straightforward in theory, but in practical way, when you work, it's not as simple. So neurostrides imaging is also extremely challenging. And then first challenge, the main challenge is if you see these structure of they are neutral molecules, that means they are not easily ionized by mass spec, and they have very similar structures, which resulted in isobaric and isomeric interferences. In addition to that, they are also present at very low concentration in our tissues, and also affected by ion separation from endogenous lipids, proteins, and molecules, which make their detection quite challenging, especially in a biological matrix such as brain tissues. So to improve this ionization and sensitivity, we develop on tissue derivatization strategy using GIRT. So GIRT is a permanently charged quaternary molecule compound that reacts with the keto functional group and under acidic condition and form this kind of a derivatized molecule, which has a pre-charge positive charge tag on it, and that's that's help ionization because it's already ion, and then that improve its ionization efficiencies and detection by mass spec. So as I said, GIT react with keto functional group. So in these all neurosteroid panel, we can select only 13 neurosteroids, which are ketosteroids that are compatible with the derivatization chemistry, and because of they have all ketonic functional group in it. Apart from cholesterol, estradiol, and androstenediol, by using this region, we can detect across the other all panels. So let me just briefly walk you through the workflow. We collected male and female P56 mice brain, snap frozen in liquid nitrogen for all kind of mass spectrometry imaging work. And then for this experiment, we done serial cross sectioning of right hemisphere at 200 micron interval. We follow the Allen Mouse Brain Reference Atlas, which provide the anatomical framework for regional annotation. And it's not like very straightforward because we need to match the histology with Brain Atlas. And also there are like other challenges, which is apart from biological variability, there are technical variability due to sample preparation of different slides and quality, which required like batch effects and other technical variability, which required robust and quality control and quality assurance measurement for reproducible and reliable results. So here we use, if you see here, you use for QC, you use a mimetic model, which is tissue homogenate, spiked with these steroid, all 13 steroid panel. And also we have a like control brain tissue, which we use for to spot all 13 mixture of 13 neurosteroids on it. And then we follow the pattern across the different samples for QA and QC procedure to pass the batch. And the next step is we spray derivatizing reagent using M5 STX M5 sprayer and incubate for an hour in humidity environment, and then we apply MALDI matrix, which helps ionization, desorption, and ionization using the same M5 sprayer. And after that, we then acquire data on the same day in FT 12 Tesla FTICRMS. We use 100 micron resolution for male and 150 for female brains. And for our data, like to see the QC, to pass the QC and QA, we process data and visually checked is fine, and then convert into ZMLs to send aspect analytics for co-registration and other like co-registration with reference atlases and other Sterol, MSI, and microscopy data. So these are just to show you the results of standard spots. So this is the mixture of 13 neurosteroids we spotted on tissue, control brain tissue, to evaluate if derivatization, to evaluate the derivatization performance, and also our optimized method is working. And we can see nice spots. So we can see very bright images of the spot on tissue, which clearly show the derivatization optimized method is working fine. And also it helps to derivatize or help to detect all 13 neurosteroids from the panel. And we also use DHB as the internal standard for normalization if needed. So, and what else we noted, there is no diffusion. We can see very compact spots shape. It means the whole process, mainly humidity, is not diffusing like steroids on the surface during the whole sample prep. And here we can see all the endogenous corticosteroid distribution across multiple sagittal brain section. As you can see, signals are not uniformly distributed throughout the brain. You can see there are the distinct pattern. It's not across everywhere. We can see the distinct pattern of corticosteroid distribution within a specific anatomical region, highlighting and suggesting the regional specificity of these neurosteroids and their regulation and signaling within the brain in a specific region. And by overlaying, so we can detect these in both male and female adult brain, and we can overlay these by Allen Mouse Brain Reference Atlas to precisely assign neurosteroid distribution to specific regions on the brain tissue. So although 13 neurosteroids were initially selected for imaging, and we are able to detect them using when you use standard spots, but for endogenous detection, we are able to detect only 6 neurosteroids consistently in brain regions, in different regions of both sexes. So it's suggested maybe other are below the limit of detection, which may require technology advances and or alternative sensitive analytical approach. And yeah, so this highlight another challenge associated with the neurosteroid imaging and importance of technology advancement and a sensitive workflow. This slide summarizes all the 6 neurosteroids. We are able to detect endogenous distribution of those in different brain region in section number 6, including corticosterone, 11-dehydrocorticosterone, 11-deoxycorticosterone, progesterone, progesterone, and estrone across the multiple brain region. Is a cortex. You can see it's in cortex, hippocampus, cerebellum, and amygdala. So these findings reveal the complexity of neurosteroid signaling across the brain region, and also demonstrate a wealth of information and wealth of data that can be extracted from a single mass spec imaging data sets or mass spec imaging run. So at this point, I would like to hand over to Bill, who will discuss more about the neurosteroid component of the project and about our web-based atlas. But before I do so, let me briefly introduce him. Bill received his BSc and PhD in chemistry from University College Cardiff, and he was trained in biomedical mass spectrometry at Karolinska Institute in Stockholm. Bill has held academic position in Kingston, Jamaica, Stockholm, Sweden, and London, England. In 2007, Bill moved to Swansea University in Wales to take the chair of mass spectrometry. Bill's research interests are in cholesterol biosynthesis and metabolism, particularly pathways of bile acids biosynthesis. Bill works closely with long-term collaborator Professor Yukin Wang, and the focus of their group work is on the involvement of cholesterol metabolites in biology, particularly in the context of rare diseases. So when living in the Caribbean, Bill played international rugby for Jamaica. Over to you, Bill.
Thanks, Shazia. So while Shazia took the neurosteroid part of this project, we've looked at the sterol. So that's really cholesterol and its precursors and its major metabolites. So cholesterol is synthesized in brain. It's synthesized by 2 pathways: the block pathway, which goes through desmosterol, and the Candace-Russell pathway, which goes through 7-dehydrocholesterol. We then get cholesterol, and the major metabolite is 24S-hydroxycholesterol. So if you look at these 4 molecules, they've all got this 3-beta hydroxy group, but none of them have got the ketone group, the 3-ketone group, or so they're not applicable to direct Girard derivatization. So we can't do a simple Girard derivatization on these molecules. However, we can get around that problem quite easily. So what we do, we take our section of mouse brain that we've cut, and then we spray on cholesterol oxidase enzyme We then incubate in a damp atmosphere, and then the 3-beta hydroxyl group of these molecules is converted into a 3-ketone group. Once we've got the 3-ketone group, we can then do the Girard derivatization. So Shazia showed you the Girard T derivative. We like to use the Girard P derivative. That's just really for historical reasons. So we should spray on our Girard reagent, incubate in a aqueous methanol acidic atmosphere, and we get the derivatization done. So we're able to increase our signals. We use for our cholesterol analysis, we use atmospheric pressure MALDI, the AP MALDI source from Mastec, and that's linked to an Orbitrap IDX. For our cholesterol precursors and cholesterol metabolites, we then use a lower resolution technique. We use the LISA technique, but we connect the LISA technique up to an LC, which is then connected to an Orbitrap. So we'll see these two techniques as the project, as the talk goes on. Okay, so let's now tell you about the atlas we've actually been building. So I should first of all say that all the funding for the whole project came from BBSRC in the UK, particularly the Bioinformatics and Biological Resource Fund, and they paid for everything. So we generated the data in Swansea and in Edinburgh. And then we collaborated with Aspect Analytics in Belgium, who then constructed the atlas. I'll tell you that the atlas is going to be released in the next 2 months, and it'll be freely available on the web for anybody to explore. So let's show you what the atlas looks like. So when you open the atlas, you get this, what we call the spatial view. So in the atlas, we've got sagittal sections of mouse brain, which are matched to the Allen mouse brain atlas. So what we actually see here is the first 4 plates of the Allen mouse brain atlas, starting at the outside, moving towards the center, and we've got all our data overlaid here on the Nissl stain of the Allen mouse brain atlas. So all our data is registered against the Nissl stain of the Allen mouse brain atlas. So in these 4 plates, we've got all our data just overlaid. And on the right-hand side, we've got what we call the control panel, where we've got all the data, and you can focus in on any particular part of that data you want. So we've got male brain, we've got female brain, we've got Shazia's neurosteroid data, we've got our cholesterol data, we've got the cholesterol precursors and the cholesterol metabolites, and they're all available here. Let's start with something easy. So let's look at the cholesterol data that we've got from using MALDI mass spectrometry imaging, and let's look at the female mouse brain. So I just click on this button here, and then I get our cholesterol data. So here we've got the first 11 panels of the Allen mouse brain, and we've overlaid our cholesterol data onto these panels. So this is the most outer panel from the Allen mouse brain atlas, and we're moving towards the midline. So each panel is separated by 200 microns. Our tissue sections are 15 microns thick, and with our MALDI here, we've got 50 micron pixels. If we just take a look at this plate, so our MALDI data, we're using a red-blue color scheme. So you can see our cholesterol MALDI data, and in it's overlaid on this Nissl stain, and this Nissl stain is this dark purple that you can see in the cerebellum. So this is the first 11 plates, and if I click on the chevron, which is up here, and then I get the next 8 plates, which takes us to the midline. Now, if you're interested in any one particular plate, you can just focus on one plate. So I'm focusing now on plate 11, and on the left-hand side, I've got our color scheme. So this is cholesterol, in our color scheme, and we go from a concentrations of essentially nothing, 0.05 nanograms per millimeter squared, up to a concentration of about 1,111 nanograms per millimeter squared. So these are really aerial densities, not concentrations. So the lowest concentration is almost certainly in this ventricle here, while we've got high concentrations in these fiber tracts. If I go down here and I hover over this text here, AMB Nissl stain microscopy, then I just get a picture of the Nissl stain from the alum mouse brain atlas. And the next slide, what I've done, I've superimposed the annotations from the alum mouse brain atlas onto our MALDI imaging. So here we got the corpus callosum, fiber tract, cerebellum, and the hindbrain. And I can click on the control panel, and I can go to the exact alum mouse brain atlas. So I've gone to this exact picture on the alum mouse brain atlas. This is plate 11 with all the Allen annotations in great detail. So what I've highlighted here in sort of purple is the fiber tracts in the alum mouse brain atlas, and you can see that's really where our cholesterol is. So the cholesterol is really high in these fiber tracts of the mouse brain. So if we're interested in one particular region of the mouse brain, I can focus on it using the scroll wheel of my mouse. So I'll focus in on this cerebellum region. So here I've got the cerebellum, and you can see we're at the pixel level here. So these are the 50 micron pixels. So we've got high levels here in the white matter tracts of the cerebellum, and then we've got lower levels of cholesterol in the gray matter. So if you see my pointer here, I'll sit on a pixel which is quite a low level of cholesterol, and then you can see that the pixel sits in the granular layer of the cerebellum. So we can match up our pixels to the alum mouse brain atlas rather nicely. So the Nissl stain really stains cell bodies rather than axons. So this is cholesterol data. We've also got Shazier's data. So this is how Shazier's data looks like in the atlas. So first of all, I should tell you that the neurosteroids are miles more difficult to work with than the cholesterol. Cholesterol is really abundant in brain. The neurosteroids are at least 1,000 times less abundant. So Shazier's got her work cut out to get such nice data. So first thing you'll notice really is that we have regions of very little neurosteroids, and these regions correspond to the fiber tracts. So we've got very low levels. This is corticosterone, these fiber tract areas. Alternatively, we've got the higher levels of corticosterone in the gray matter of the cerebellum. So again, if you watch my pointer, and I'm going onto a high level of corticosterone, when I show the Nissl stain, that's bang in the granular layer. So in cell bodies, we've got high levels of corticosterone. Well, alternatively, the cholesterol was focused in the fiber tracts, and I can actually overlay our cholesterol data onto Shazier's neurosteroid data. So our cholesterol data is now in white, and you can see the cholesterol in the fiber tracts, while Shazier's neurosteroids, as I said, is much more in the grey matter. So this is a nice illustration how we're matching Shazier's neurosteroids against our cholesterol, all matched all registered against the alum mouse brain Nissl stain. Okay, let's move on. That was cholesterol and the neurosteroids. Now let's look at the cholesterol precursors. As I mentioned earlier, there are two pathways of cholesterol biosynthesis. There's the block pathway, which goes through desmosterol. So desmosterol is just like cholesterol, but it's got an extra double bond in the side chain. And then the Candice-Russell pathway goes through 7-dehydrocholesterol, which is again like cholesterol, but it's got an extra double bond in the B ring. So these cholesterol precursors, they're a bit like the neurosteroids. They're about 1,000 times lower abundance than cholesterol. So we've got an analytical challenge in terms of low abundance, and also you can see their isomers. So we've got a problem here because we want to be able to resolve these isomers, and that's a problem if you're going to use MALDI. So we decided to switch techniques, and we decided to use ELISA. So ELISA stands for liquid extraction for surface analysis. So here we have essentially spot extraction of the tissue. So what we do, first step is we do prepare our sample in exactly the same way as for MALDI, except we don't need now to bother with the matrix. We then create a sort of a grid template where we split our brain into 800 micron pixel size. So that's the size of the resolution that we can get with the ELISA. So then the ELISA is as I said, is a liquid extraction for surface analysis. So basically we have a robotic probe that goes down onto the tissue, and we have solvent in the robotic probe which extracts the sterols. The sterols are then extracted, and we come up here. We've then got the probe is then connected to an LC. So the extracted sterols then go through this rather convoluted plumbing system, and they first the sterols are trapped on a trap column, C18 trap column, and we wash away the Girard reagent. We then elute the sterols from the trap column onto an analytical column, and here we're able to separate out the different sterols. So we're able to separate out these isomers. So the advantage of using the ELISA is we can connect it to an LC, and we can separate out isomers. The disadvantage is, of course, is that really major loss in resolution. So let's now show you some ELISA data. So this is desmosterol, and we've got the same color scheme as before. Dark red is abundant. Dark blue is of low abundance. So first thing to note is, look on my left-hand column here, is that the aerial densities of desmosterol are much lower than cholesterol. So cholesterol, it was about 1,000 nanograms per millimeter squared. With desmosterol, we're down to 5 nanograms per millimeter squared. So it's at least 200 times lower abundance. So you can see there's very little desmosterol in the isocortex and the cortex of the cerebellum, but you have good levels of desmosterol in the fiber tract regions, and also good level in the hindbrain. Okay, that's the cholesterol precursor. I just showed you the desmosterol data, but we can generate similar data for 7-dehydrocholesterol. Let's now move on to cholesterol metabolites. So the major cholesterol metabolite in brain is this one, 24S hydroxycholesterol, and where we've got an added hydroxyl group on position 24 of the cholesterol side chain. So this is the major cholesterol metabolite that we find in brain. We also find other cholesterol metabolites, such as this one, which is 27 hydroxycholesterol. So here you've got a hydroxyl group added on to position 27. But this is more minor than 24 hydroxycholesterol. A problem with cholesterol and cholesterol metabolism and mass spectrometry imaging is that we slice our brain samples in an air atmosphere. So when you slice your brain, you cut your brain tissue, it's immediately exposed to air. And when you expose cholesterol to air, you've got the possibility of what we call auto-oxidation. That means cholesterol can be oxidized by the oxygen in the air. So when you have cholesterol and air, you get this epoxide formed, which is cholesterol 5-6 epoxide. And the cholesterol 5-6 epoxide, actually in the derivatization, will isomerize to this, which is 6-beta hydroxycholesterol. So So we've got a rather complicated pattern here in that we've got 27 hydroxycholesterol, 24 hydroxycholesterol, which is the major metabolite, and we've got these two others, which are auto-oxidation artifacts, and in fact, and they're all isomers. So this is where the LISA, where the LC comes in. So we're able to separate out all these different isomers. So we can measure the 24S hydroxycholesterol accurately without contamination of the auto-oxidation artifacts or 27 hydroxycholesterol. So let's now show you the LISA data for 24S hydroxycholesterol. So 24S hydroxycholesterol. Again, if you look at the left-hand corner here, again, the concentrations compared with cholesterol are really low. So remember, cholesterol got up to 1,000 nanograms per millimeter squared. Here, 24-hydroxycholesterol were down to 2 nanograms per millimeter squared. So again, it's just illustrating that we have an analytical challenge in terms of sensitivity here. So most of the 24-hydroxycholesterol is in the striatum and the thalamus and pallidum. There's very little 24S hydroxycholesterol in the cerebellum and very little in the hindbrain. So I can now overlay my cholesterol data onto the 24S hydroxycholesterol. So the cholesterol is in white, and the 24-hydroxycholesterol, the most abundant, is in red. So you can see, if you just look at the hindbrain here, although you've got plenty of cholesterol, you've got almost no 24-hydroxycholesterol. The same is true in the cerebellum. The 24-hydroxycholesterol, it really is focused in the striatum and thalamus. So this is quite nice, really, because you can see that there's the concentration of product does not necessarily correlate with the concentration of substrate. The substrate here is abundant, but there's no product. That tells us the reason for that, of course, is there's no enzyme, there's no CYP46A1 in this particular region. Okay, so all the data that I've shown you on our atlas is what we call spatial view. We can also see all the data in what we call plot view. So here I've plotted the different regions of the Allen mouse brain atlas against the areal density of 24-hydroxycholesterol. But I could do that for any of the different analytes that we analyze in our experiment. So we can also have the neurosteroids here or cholesterol, whatever you like. So certainly for 24S hydroxycholesterol, here you see the highest levels of striatum in pallidum and much lower levels in hindbrain. So this is actually all our data here. So this is interquartiles, and this is the extreme data point of or the extreme pixel point. So that's what we call the plot view. In fact, if you click here, you get what we call the table view, and this is where you've got all the data in numbers. So here we've got, again, I'm just focusing on 24-hydroxycholesterol here, but I could do the same for all the different analytes. So 24-hydroxycholesterol, this is the mean value for each different region. So it's most abundant in the pallidum and striatum. Or if I want to look at the median, here are my median values. Here are my interquartile values, or 95 and 5 percentiles. So basically, all the data will be made available for all the different metabolites for you to look at and enjoy it at your pleasure. So finally, a final slide. So in Edinburgh, the work done by Shazia with supervision of Joyce Yao. Ruth Andrew is the co-PI on the project. In Swansea, the co-PI is Yutin Wong, and then Elan did a lot of the work. Elan's moved, and now it's Tom Ginges Perez. Moses also been a lot of help with the Girard derivatization, and the people who built Atlas Aspect Analytics. So that's Heath Patterson, Tao Tran, Wang Qin Zhang, Nico Verbeek, Alice Lee, and Mark Klassen. So Mark was a great help at the beginning of the project as we were very slow in getting it off the ground in terms of passing the funding to aspects. So Mark was been a great help, and of course the funders BBSRC. We got a little bit of money from the Welsh Government and then Edinburgh University also. So as I said, the Atlas will be released now in the next couple of months. And what's holding us back at the moment is ourselves, really, because we're trying to write a manual to describe how to use it, and that's really our current project that's holding us up. But when it's when that's completed, it'll be made publicly available. So if you want to drop me an email to this address, saying you want access to the Atlas, then I'll make sure that as soon as it's made public, I send you the link to have a look. So I think that's more or less it. So I think we can take the questions and answers. So Jennifer, would you like to ask your question?
Certainly. Thank you so much. Great talk. I was just curious as to why you chose to use 100 micron for the males and 150 micron for the females.
Yeah, I think good question. You spotted that. So initially, actually, we started from 150 micron because the level if we go to the high resolution, we reduce the surface analysis. There is a trade-off, like less signals if we go high resolution. So initially we thought we can go for 150 micron, and we collected all female data, and then we tested 100 micron. We still got very good signals in 100 micron. So so then for male experiment, we collected 100 micron. We try lower than that, like 75 micron and 50, but then we compromise the signal intensities. So because of the neurosteroids are not very abundant on surface. So so that was just the case.
Perfect. Thank you so much.
So next question from Jens. I can read out Jens's question. Seems to measure the steroid hormones from gonads as the distribution of progesterone. How do you determine brain-specific products? You talked about neurosteroids. Did you mean steroids distributed to the brain? I guess this is a question everyone asks about neurosteroids, Shazia.
Mm-hmm. Yeah. So I think this reflects the enzyme local activity in these specific region and production from cholesterol and then gray matters. So there are, it has been noted that there are more enzyme expression in these regions, which reflect the steroid distribution in the same place. So that's so specific metabolism is going on this.
Jens, are you there? So do you want to ask your second question?
So yes, in addition to what you said, this shows endogenous production in the cortex region of the brain where you have high progesterone. There is no production of progesterone. That must be from external. That's one comment. And this question that I had, the Atlas shows steroid in the white substance, which is the connections in the brain, which is approximately 80% of fat. How about the hypothalamus region where you actually have endogenous production? How does it look there? Do you have any example of this?
Yeah, I think we didn't dig out much on this, but there must be enzymatic activity in these regions. So that's we need to co-register with other data sets to see what's going on in maybe neurotransmitter systems there, or so more. That's my thought. Maybe there's no more attached to the neurons or synaptic activities and this kind of stuff. Yeah.
Okay. Next question, Robbie.
Hi, Saria. Thank you very much for your nice presentation. I have a few questions, like which metrics you use and which composition you use after derivatization.
Yes. So for me, I use CHCA, which is quite common matrix. And positive mode.
Okay. So you use the 10 mg per ml or 15 mg per ml?
10 mg per ml in acetonitrile water, 50% and 0.2% TFA.
Okay. Hello, Ed.
A great talk. I have a question about cholesterol. Is there much cholesterol ester or can you measure it
That's
in brain?
that's a very, very good question. So the traditional dogma is that the cholesterol esters are probably present at about 1% of cholesterol, but that's traditional dogma. But it seems that there's evidence now in a number of the neurodegenerative diseases that the levels of cholesterol esters are higher. So yeah, for our method, it's obviously we can't do the simple derivatization on the cholesterol esters, but what we could do is to actually spray a esterase enzyme and then look at the change in levels of cholesterol with and without the esterase enzyme. But that's not such a beautiful way to go. So I think cholesterol esters is probably worth investigating, but probably better not to use our derivatization. Maybe find a different way to look at them. I definitely think cholesterol esters. We're certainly worrying about that. Whether we're missing interesting stuff.
The enzyme would be difficult to do and expect it to work universally that way as a spray. How about a little base?
Yeah.
Or
Yeah,
acid slightly buffered that would be less discriminatory. You might get a lot of other stuff, but you might see some change, or together with a derivatization of the esterified product.
Yeah, that's probably a good idea, right? We may well try that, Ed. I think that is I guess that's in our mind, but we haven't. But it's not the front of our mind. So that's something we'll mark down as an idea to explore. So thanks for that. Thanks for that suggestion.
Okay, thank you.
Okay, next one is from Clarissa.
So, did you see anything specific in the ventricles?
No, so we didn't really, we haven't looked for the cholesterol esters, and there's essentially no cholesterol in the ventricles. I mean, no free cholesterol in the ventricles.
And I was wondering because because to make the cholesterol ester, you need the cholesterol.
Sure.
And to make also the derivatives that you found, like the oxidized cholesterol. So would it be there's a transport of where is it formed, this oxidized cholesterol or this products that you found?
Okay.
Because you have to have the cholesterol there in order to be able to make it or transport oxidize it, that I don't believe it will.
Yeah. So I guess in our figures the imaging figures are really quite biased to the eye. So there's, other than the ventricle, there's cholesterol everywhere. And actually, the highest levels of the cholesterol metabolite, that's 24S-hydroxycholesterol, they really correlate with the presence of the enzyme, the CYP46A1 enzyme. So you can really match the enzyme expression to the level of that 24S-hydroxycholesterol. So it's there's everywhere except that ventricle there is cholesterol. It's just the levels are lower than in those really eye-catching fiber tracts. So I guess this is imaging can be quite distorting. Our eyes can distort the truth.
Yeah. So this was also one of the question that I posted. That have you crossed with the protein expression maps?
Oh yeah,
that are already there, and then you can find out the.
That's a very, very good question. That's something that we could do in essentially a next version of the next and in a next or upgraded version of the atlas, because the Allen Mouse Brain Atlas, does have expression of lots of different proteins. So it would be theoretically possible to match our data to the Allen expression data, but that would be another project that we'd have to fund aspect to do. But I mean, but that's something that should be done if there was a funder to fund it. I can ask Yannick's question. So does the cholesterol I get from my diet or the cholesterol levels on my regular blood test have any impact on this brain map? No, is the answer, because we have what's called the blood-brain barrier, and this isolates the brain from cholesterol in the bloodstream. So cholesterol in the bloodstream will not cross into the brain. So our brain is sort of isolated from our bloodstream. So you don't need to worry about your high cholesterol levels in the blood in terms of the cholesterol itself getting into the brain. However, cholesterol metabolites, because they've got an extra functional group, like if they have an extra hydroxyl group, they can actually cross the blood-brain barrier and get into the brain. So maybe you do also have to. So you still have to worry about your high cholesterol levels. And I see Jeff McDonald from Dallas. Is there, Jeff? Would you like to ask your question?
Yeah, please. Back up, follow for great presentation, great work. Following up on Ed's inquiry on the cholesterol ester. If you image it or if you take a sample, can you comment on, is it actually just in the circulating periphery, back to the blood-brain barrier issue, and you're just sampling the blood in the tissue, or do you know, are the cholesterol esters are they being deposited in the tissues or the neurons or the structural components of the brain?
Yeah, that again. That's a good question, because I guess there's always this worry about what you're measuring, whether it's whether it's actually in the blood vessels. So that's a good question, and the answer is, I guess we don't know. But that's something that in these brain imaging experiments, you always have to be aware that something that you're seeing is from the blood vessels. So yeah, that's a good question, and the answer is I don't know. No, I think that's it. Okay, so thank you everybody for listening to the seminar. Okay, thank you everybody for watching, and
Thank you. Bye bye.
Bye bye.